Welcome to the Longevity Papers podcast.
We're back again. And for all of you out there, the biotech researchers, VCs, the folks actually doing the lab work.
Well, our job here is to help cut through the noise.
Exactly. We want to help you figure out that signal-to-noise ratio in the flood of papers that come out each week.
Are we looking at just small, incremental steps? Or is there something genuinely, model changing that you really need to pay attention to?
Yeah, because you're busy. Running assays, designing the next trial, maybe fighting for funding. You don't necessarily have time to read every single PDF that lands.
Especially when, let's be honest, most weeks, maybe 95% of the published research, it's solid, it's necessary science, but it's often refining a technique or maybe an observational study. It pushes things forward, but slowly.
Right. It's not bending the curve, so to speak.
And that's our mandatory dose of skepticism right up front. This week, October 13th to the 19th, 2025, it's pretty much a textbook example of that.
A typical week. A very typical week. We saw some really good incremental work, data atlases, some big observational studies, biomarker correlations, all valuable stuff.
But no magic bullet.
Definitely no paper showing, a 30% lifespan expansion popping up out of nowhere in a complex model. It's foundational work this week, not revolutionary. Well, perhaps with one exception in terms of technique.
Okay, so let's dig in and find that signal. We've picked out 5 papers we think are worth your time.
And we'll structure it, starting with what we think has the highest novelty, then move towards the more foundational, data-heavy stuff.
Sounds good. So first up is a really interesting gene editing paper for Progeria. Seems like a potential new approach.
Yes, that one definitely stood out. Then we'll get into some solid mechanistic work on chaperone-mediated autophagy, CMA, with some surprising sex-specific findings in neurobiology.
Okay.
And finally, we'll tackle those big data papers, a huge multi-omics aging clock effort, and a really impressive spatial transcriptomic atlas of the human cortex. Lots of data there.
Right, the kind of papers that are resources but need careful interpretation.
Exactly. So let's start with what feels like the most genuinely innovative technique this week. It's a potential shift in how we think about editing for certain diseases. That's the Progeria paper from the team in Korea.
All right, let's dive into that one.
So the paper title is Mutation-Agnostic Base Editing of the Progerin Farnesylation Site Rescues Hutchinson-Gilford Progeria Syndrome Phenotypes in Neuromuscular Organoids.
Got it. And this came out quick, October 16th on bioRxiv.
Right.
Pronounced bioRxiv, remember?
Right, bioRxiv. And it's from Kim, Kwan, and Jian at the College of Pharmacy, Seoul National University.
Okay, so HGPS, Hutchinson-Gilford Progeria Syndrome. It's just a devastating disease, right? Rapid aging in children.
Absolutely devastating. It's caused by this toxic protein, progerin, which is a mutated form of lamin A that gets improperly farnesylated. Farnesylation is a lipid modification.
And that messes up the whole structure of the cell nucleus.
Completely. It causes the nuclear envelope to sort of bleb and become unstable, leading to massive problems with DNA repair, senescence, basically accelerated aging at the cellular level.
And current treatments like farnesyltransferase inhibitors.
Mm-hmm.
They help a bit, but not much.
Only modestly. The problem is they inhibit all farnesylation in the body, not just the abnormal progerin. So you get off-target effects, and the benefit is limited.
Okay, so lack of specificity. And trying to fix the specific gene mutation with CRISPR is also tricky.
Very tricky. Because while there's one common mutation, G608G, there are actually quite a few other rare mutations in the element A gene that can also lead to these progerin-like toxic proteins. We call them atypical laminopathies.
So you'd need a different gene editor for potentially dozens of different mutations, which isn't practical for developing a therapy.
Exactly. And that's the beauty of what this Seoul National team did. They developed something called FATE, farnesylation amino acid targeted editing.
FATEC.
Okay.
And the key here is that it's mutation agnostic. It doesn't care exactly what the underlying DNA typo is. So how does it work then, if it's not fixing the specific mutation?
It uses base editing, which is a more precise form of CRISPR, to target the code for the farnesylation site itself within the element A gene transcript. It essentially edits the instructions so that the final lamin A protein simply cannot be farnesylated.
Ah, I see. So it prevents the toxic modification from happening in the first place, regardless of the original mutation that caused the problem.
Precisely. It disrupts that specific element A farnesylation signal, but it's designed not to affect the normal farnesylation of other essential proteins in the cell. It targets the root mechanism of the disease pathology.
That does sound like a potential paradigm shift.
Yeah.
Very clever. So did it work in their model? They used neuromuscular organoids, right? NMOs.
Yes, isogenic human pluripotent stem cell-derived NMOs. A relevant model because muscle and vascular problems are big in HGPS. And the rescue at the cellular level looked pretty impressive.
What did they see?
Well, first, the FATE treatment got rid of that characteristic paranuclear progerin accumulation. The nuclear shape normalized. That's a big visual confirmation.
Yeah.
And functionally, they showed it restored the mobility of 53BP1, which is crucial for efficient DNA repair.
Right, that's a key marker.
It allowed for the proper formation of DNA repair foci, and it seemed to normalize the messy heterochromatin architecture you see in these progeroid cells. Basically, key cellular hallmarks of aging seemed reversed in these organoids.
And how did they deliver the FATE system?
They used transient delivery, mRNA encoding the FATE base editor, packaged into lipid nanoparticles, LNPs, and delivered directly to the organoids.
Okay, so LNP delivery. That points towards a potential therapeutic path using RNA.
Yeah, it's a proof of concept for that approach, definitely.
This sounds really promising. Technically elegant, targets the core problem, mutation agnostic. So, okay, time for the skepticism. Why isn't this a slam dunk yet for a biotech team looking at it? The caveats must be significant.
Oh, absolutely. The caveats are huge. And it really comes down to the vast difference between fixing cells in a dish, even complex organoids, and treating a whole patient with a systemic disease like HGPS.
Okay, caveat number one, the model itself.
Exactly. NMOs are great for looking at neuromuscular junctions, but HGPS is multi-system. And critically, the primary cause of death in these children is cardiovascular disease, severe early onset atherosclerosis, hardening of the arteries.
And this paper didn't show any effect on vascular cells or cardiovascular function?
Not in this preprint. They should rescue in NMOs, but fixing muscle cells doesn't tell you if you fix the aorta. You absolutely need to show this works in relevant cardiovascular cell types, like vascular smooth muscle cells, or ideally, in an HGPS mouse model, looking at cardiovascular outcomes. Without that, the therapeutic claim is, well, incomplete.
Right, that's a major gap. What else? Delivery.
Delivery is always a hurdle for in vivo gene editing. LNPs are the state-of-the-art for RNA delivery, yes, but scaling that up for systemic delivery, especially potentially needing repeat dosing for a chronic condition, that brings massive challenges.
Like liver toxicity, off-target delivery, immune responses.
All of the above. We're still figuring out the long-term safety profile and efficiency of systemic LNP delivery, especially with repeated administration. It's a major bottleneck for the whole field, not just this specific therapy.
And it's a bioarchive preprint, so not yet peer-reviewed.
Correct. And organoid studies, while powerful, often use smaller sample sizes. You need independent replication. Plus, we need comprehensive, unbiased long-term assessment of off-target editing. Base editors are precise, much more so than older CRISPR-Cas9, but they're not perfect. We need to be sure it's only editing the intended site in the LMNA gene and not causing problems elsewhere in the genome over time.
Okay, so the bottom line for someone evaluating this.
It's a genuinely exciting platform innovation, a new strategy, but the required next steps are crystal clear and absolutely non-negotiable before you'd invest heavily.
You need in vivo validation in HGPS mouse models, specifically showing improved cardiovascular health and lifespan.
And rigorous, deep sequencing-based off-target analysis across multiple tissues over the long term.
Until then, it's a brilliant idea for how to edit, but we don't know yet if that editing actually cures the patient systemically and safely.
Precisely. High potential, high hurdles remain.
Okay, that makes sense. A promising but early-stage technology. Let's shift gears now from a very specific, rare genetic disease to the broader challenge of aging in the brain.
Right. Moving to paper 4, this one's in a high-impact journal, Nature Cell Biology, published October 15th.
The title is Chaperone-Mediated Autophagy Regulates Neuronal Activity by Sex-Specific Remodeling of the Synaptic Proteome. Authors are Kawaja, Grigo, Lindenau, and colleagues from Albert Einstein College of Medicine.
So this is about chaperone-mediated autophagy, CMA. Think of it as one of the cell's key quality control systems. It identifies specific soluble proteins that are damaged or no longer needed and targets them for degradation in the lysosome.
And we know CMA function goes down with age, right? And it's implicated in things like Alzheimer's and Parkinson's.
Exactly, the well-established link. CMA decline is seen in aging across tissues, and specifically losing CMA function just in neurons is known to cause neurodegeneration, at least in mouse models.
So what's the core finding of this new paper?
They established a pretty direct causal link. If you knock down CMA in neurons, you get neuronal hyperactivity. The neurons become overly excitable.
Like they're firing too much or too easily?
Yes, increased seizure susceptibility, problems with calcium balance. Basically, the neurons are stressed and malfunctioning because they can't clear out the junk proteins properly.
Okay, that makes intuitive sense. Breakdown in quality control leads to malfunction. But what's the really novel part here?
The novelty, and this is really the key takeaway, is the striking sexual dimorphism they found in how CMA regulates the synapse.
Sexual dimorphism, meaning it works differently in male versus female neurons.
Fundamentally differently, at the level of the synapse. They looked at the synaptic proteome, all the proteins at the connection point between neurons, and found CMA was remodeling different parts in males versus females.
Wow. Okay, break that down. What was the difference?
In the female mice lacking proper CMA function, the main problem seemed to be at the presynaptic terminal. They saw enhanced neurotransmitter release, issues with NMDA receptor transmission. It looked like the machinery for sending the signal was dysregulated. CMA normally keeps that presynaptic proteome in check in females.
Okay, so presynaptic in females, what about the males?
In the males, the dysfunction was predominantly postsynaptic. They showed elevated activity of AMPA receptors, which are key proteins on the receiving side of the synapse. It seems in males, CMA preferentially targets and remodels proteins in the postsynaptic compartment.
That is really quite profound. The same basic cleaning system, CMA, is tidying up different parts of the synapse depending on sex.
Exactly. It implies that the way synapses might decline with age, or in neurodegenerative disease where CMA is impaired, could follow fundamentally different molecular paths in males versus females.
That has huge implications for drug development, doesn't it? If you develop a drug targeting, say, a postsynaptic protein that CMA normally degrades.
It might work great in males, but do absolutely nothing in females, or vice versa, if you target a presynaptic component.
It really underscores the need to consider sex as a biological variable at the most fundamental mechanistic level.
It really does. It makes you question how many potentially sex-specific drugs have failed in mixed-sex trials because the effect was diluted or averaged out.
Okay, so that's the core finding. Did they show any therapeutic potential? Could boosting CMA help?
Yes, that was the encouraging part. They showed that either genetically reactivating CMA or using a pharmacological CMA activator compound.
Oh, they have a compound.
They used a known experimental CMA activator. And in both old mice and an Alzheimer's disease mouse model, activating CMA helped. It restored synaptic protein levels towards normal, it reduced that neuronal hyperexcitability and seizure susceptibility, and it seemed to normalize neurotransmission patterns.
So it suggests CMA is indeed a druggable target for age-related neuronal dysfunction or potentially AD.
The potential is certainly there. It's strong preclinical evidence.
All right, critical analysis time. What are the translational hurdles here, beyond the usual mouse-to-human caveats?
Well, the mouse-to-human gap is always present, especially with AD models. Many AD mouse models are based heavily on amyloid overexpression, and they don't always capture the full complexity of human AD, particularly the tau pathology or the subtle cognitive decline.
Right. And did they show actual cognitive improvement? Or just normalization of electrical activity?
That's a key point.
Yeah.
The paper focused more on the electrophysiology, the hyperactivity and seizure risk. While that's important, it's not the same as demonstrating improved memory or learning. For longevity or AD therapy, you ultimately need to show cognitive benefits, not just quieter neurons.
Good point. We need behavioral validation. What about the target itself? CMA is fundamental. Any risks to boosting it long term?
That's the other major concern. CMA is essential housekeeping. If you chronically activate it systemically with a drug, what are the potential unintended consequences?
Like it starts breaking down proteins that are actually needed. Overcleaning.
Potentially, yes. You might disrupt the normal turnover of essential proteins or affect other pathways that rely on CMA substrates. It's a fundamental catabolic process. Long-term, systemic activation needs very careful evaluation for safety. Years-long safety studies would be needed to ensure you're not causing problems elsewhere by pushing the system too hard for too long.
Okay. So, strong mechanistic insight, clear therapeutic potential, but definite hurdles around cognitive validation and the long-term safety of activating such a core process.
That sums it up well. Fascinating biology, especially the sex difference.
Definitely. Okay, let's switch gears again. From the brain to the bones, tissue repair is crucial for health span.
Indeed. Paper 5 takes us into macrophage polarization and bone healing and aging.
This one is accelerated regeneration of senescent bone injury through age and sex-independent macrophage polarization. from Fukuda, Takamaru, Kang, and others at Tokushima University Graduate School. Published October 15th in ACS Applied Materials and Interfaces.
Right, so this tackles a really common problem. Why do injuries, especially bone injuries, heal so much slower as we get older?
And a big part of that is inflammation, right? Chronic inflammation or inflammaging.
Exactly. And specifically, it involves immune cells called macrophages. When you get injured, macrophages rush in. Initially, they need to be in a pro-inflammatory state called M1 to clear debris and fight infection.
But then they need to switch.
They absolutely need to switch to an anti-inflammatory pro-healing state called M2. These M2 macrophages release factors that promote tissue repair, blood vessel growth, and tell other cells to rebuild.
And in older individuals, that switch gets stuck. They stay to M1 for too long.
Say precisely. The transition is impaired. They remain in that inflammatory M1 state, which actually inhibits healing and contributes to chronic inflammation. This is a major reason why fractures don't heal well in the elderly, and why some existing therapies that try to boost healing factors don't work as well in older people.
Okay, so the Tokushima team proposed a way to force that switch.
Yes, using a clever nanomedicine approach. Phosphatidylserine liposomes, or PSLs.
Phosphatidylserine, isn't that the lipid that gets exposed on cells undergoing apoptosis, cell death?
Exactly. It's like an eat-me signal for macrophages, telling them to clean up peacefully. The idea here is that these PSLs mimic apoptotic cells and gently nudge the macrophages toward that anti-inflammatory pro-resolving M2 state.
And the key finding?
The really striking finding was that these PSLs induced M2 polarization regardless of age or sex.
Okay, that's interesting. Age and sex independent.
Yes. They compared young mice, 3 to 4 months, to genuinely old mice, over 21 months, and saw the same pro-M2 switching effect in macrophages from both males and females.
That potentially bypasses a lot of the complexity of age-related inflammation and hormonal differences. That sounds quite valuable.
It is. It suggests they tapped into a pretty fundamental pathway for macrophage polarization.
So how does this relate specifically to bone healing? M1 versus M2 and bone cells?
They showed mechanistically what we generally know. Factors secreted by those pro-inflammatory N1 macrophages directly inhibit osteoblasts, the cells that build new bone.
Right.
And M1 factors enhance osteoclasts, the cells that break down bone. So M1 macrophages tilt the balance towards bone destruction, and this bad effect was even more pronounced with macrophages taken from older mice.
Okay, and the M2 macrophages induced by the PSLs did the opposite.
Yes, M2 macrophages promote osteoblast function and suppress osteoclasts, shifting the balance towards bone formation and repair.
And did this work in vivo, in an actual bone injury model?
It did. They used a critical-sized bone defect model, in old mice basically, a hole in the bone that wouldn't normally heal on its own. When they treated these old mice with PSLs locally at the injury site,
They saw better healing.
significantly better. The PSLs induced the M1 to M2 switch in the macrophages within the wound, improved that crucial osteoblast-osteoclast balance, and led to accelerated bone regeneration, almost comparable to what you'd see in young mice.
Wow. Okay, so that sounds clinically very relevant, especially for orthopedics, maybe even dentistry, dealing with bone loss in older patients.
Definitely high potential value there. And the age and sex independence is a huge plus, simplifying potential clinical trial design.
All right, limitations. What are the caveats here?
Well, one is that the whole M1, M2 macrophage paradigm, while useful, is increasingly seen by immunologists as a bit of an oversimplification.
Right, it's more of a spectrum.
Exactly. Macrophage polarization is incredibly complex and context-dependent, influenced by dozens of signals in the local tissue microenvironment. These PSLs worked in the specific context of a bone defect. Would they work the same way in, say, muscle tissue or skin or the brain, where the signaling environment is totally different? We don't know yet.
So it might be a niche effect for bone.
It could be. We need to see if this approach is robust enough to push macrophages towards M2 in other tissues with different inflammatory milieus.
And what about the delivery? They used local application here.
Yes, local injection into the bone defect. For broader applications, like maybe treating systemic inflammation or promoting repair in multiple tissues, you'd need to think about systemic delivery. And then you run into the usual challenges with liposomes.
Bioavailability, stability, getting them to the right place without being cleared too quickly.
All of that. Maintaining the integrity and concentration of PSL systemically is a significant hurdle if the goal moves beyond localized treatment.
So future directions. Needs testing in other tissues.
Absolutely. To be truly impactful for broad longevity, they need to show this works for muscle regeneration, skin healing, maybe even fibrosis in organs. Right now, it's a very promising, elegant solution for a specific, important age-related problem bone repair.
Got it. Okay, let's shift from interventions to the big data landscape, observational studies.
Right. These are the papers that generate massive data sets, often acting as resources for the whole field. But extracting clear, causal, actionable insights can be really tough.
The correlation versus causation problem looms large here.
Always. So we have two big ones this week.
First is a multi-omics aging clock paper.
Yeah, paper two. Phenom-wide multi-omics integration uncovers distinct archetypes of human aging. This was posted October 14th on ArcSoar. Authors are Li, Tang, Xu, and a large consortium.
Okay, phenom-wide multi-omics integration. Sounds ambitious.
Extremely ambitious. They use data from the Human Phenotype Project cohort. That's about 12,000 adults aged 30 to 70, followed longitudinally.
And they threw everything at it.
Pretty much. Clinical data, behavior, environment, plus multiple omics layers. Transcriptomics, lipidomics, metabolomics, and the microbiome, both gut and oral.
And they used machine learning to build aging clocks.
Yes, advanced ML like GBM specifically, to integrate all this data and build clocks that predict health outcomes, and also to look for patterns, or what they call archetypes of aging.
Okay, so what are the main claims or findings we should pay attention to?
Claim one is that their multi-omics clock predicts health outcomes better than clocks based on just one data type, which isn't too surprising but good to confirm. Claim two is the more interesting one. They say they found distinct biological subtypes or archetypes of how people age based on clustering these multi-omics profiles.
Suggesting there isn't just one way to age faster or slower.
Exactly. Let's pull out a couple of key findings from their analysis. Finding one really highlights the microbiome again.
Seems to be a recurring theme.
It really is. Their aging acceleration scores derived just from the gut and oral microbiome data showed the strongest and broadest associations with all sorts of physiological systems. Frailty, liver health, insulin resistance, you name it.
So the microbiome reflects overall systemic health really well.
It seems to. Those microbiome scores were linked to 14 out of the 16 non-cancer diseases they tracked. That makes them look like powerful indicators of general health and risk for multiple age-related diseases.
Okay, what else? The aging trajectories.
Finding two relates to the pattern of accelerated aging. They argue it's not just a steady, faster decline. Instead, it's marked by a distinct, large-scale disruption across multiple omics layers that seems to kick off relatively early, around midlife, maybe age 50 or so.
A midlife crisis at the molecular level.
Kind of, and they link this disruption strongly to that microbiome axis we just talked about. Conversely, people aging slower showed prolonged stability in their molecular profiles. It suggests a potential critical window for intervention around midlife.
And finding three, did they see sex differences here too?
They did, and it aligns remarkably well with the CMA paper we discussed earlier. In the group classified as accelerated agers, the females showed a really sharp wave of molecular changes peaking around 50 to 55 years old.
Right around menopause.
Exactly. While the males in the accelerated group showed a more drawn-out, delayed wave of changes that peak maybe 10 or 15 years later.
That's a really compelling observational evidence for different aging timelines and patterns between the sexes.
It really is. Reinforces that sex needs to be considered.
Okay, so lots of interesting correlations and patterns, but skepticism time. What are the major critiques of this kind of big data clock and archetype study?
Well, the claim of finding distinct archetypes needs to be taken with a grain of salt. When you apply powerful, unsupervised clustering algorithms like GBM to incredibly high-dimensional data like this.
You're almost guaranteed to find some clusters, whether they're biologically meaningful or not.
Pretty much. These clusters could just be artifacts of the data structure, the specific algorithms used, or even noise in the measurements, especially with the massive multiple testing burden across thousands of features and multiple omics layers. Replication in completely independent cohorts is absolutely essential before we accept these archetypes as real biological entities.
Fair enough. Pattern recognition can find patterns even in randomness sometimes. What about the age range, 30 to 70?
That's another limitation for longevity research specifically. By stopping at 70, they're missing the period where the most dramatic age-related physiological decline in mortality occurs. This is really a study of midlife health trajectories and risk factors, not necessarily the determinants of reaching extreme old age.
So it's an incremental advance over existing clocks, like the Horvath or epigenetic clocks.
It adds more data layers,
Yeah.
particularly the microbiome, which seems important. But the fundamental challenge remains. These are correlations. This study tells us what accelerated aging looks like across multiple omics layers, especially concerning the microbiome and timing differences between sexes.
But it doesn't tell us why it's happening or what specific molecule to target to change that trajectory.
Exactly. It provides potential biomarkers, maybe helps stratify patients for trials based on these archetypes if they replicate, but it doesn't give you a direct causal drug target. That still requires mechanistic work.
Makes sense. Useful resource, but not a therapeutic roadmap. All right, one more resource paper. This one sounds technically incredible. The Spatial Atlas of the Cortex.
Yes, paper 3, a population-scale single-cell spatial transcriptomic atlas of the human cortex. Also out October 14th on bioRxiv from Han, Liu, Wang, and the team at Linging Laboratory in Shanghai.
Population scale, single cell, spatial transcriptomics. That's combining several cutting-edge technologies.
It's a technical tour de force. They generated this atlas from 71 human brain donors, spanning the lifespan. Spatial transcriptomics lets you see not just what genes are expressed in each cell, but where that cell is located within the brain tissue architecture.
So you get the gene expression map overlaid onto the physical structure.
Exactly.
Yeah.
Hugely powerful for understanding how different cell types are organized and interact in specific brain layers. They identified over 900 genes with layer-specific expression patterns. That alone is a valuable resource for neuroscientists.
And what did they find specifically related to aging in the cortex?
Well, their findings largely confirmed what we already know are the major hallmarks of brain aging, but now with much higher spatial resolution.
Such as?
They saw evidence of neuronal vulnerability, particularly a decline in certain types of inhibitory neurons, SST neurons, in the superficial layers. And they saw widespread glial activation, microglia, and astrocytes becoming more reactive, which is a classic sign of aging and inflammation in the brain.
They also noted changes in cell communication.
Yes, they highlighted enhanced communication between neurons and glial cells, specifically mentioning the neuregulin signaling pathway as being more active in the aged cortex.
Okay, so technically amazing, beautiful data confirms known biology with spatial detail. But for the biotech researcher looking for a new target, what's the limitation here? It feels like a detailed map of a known territory.
That's a good analogy. The primary limitation is that it's an observational study using post-mortem human tissue.
So dead brains.
Dead brains. Which means you absolutely cannot distinguish cause from effect. Are the glia activated because the neurons nearby are dying or stressed? Or is the glial activation itself driving the neuronal dysfunction? This data can't tell you that. It's a snapshot in time at the end of life.
And the main findings, like glial activation and neuronal loss, are already well-established concepts in brain aging.
They are. This study provides fantastic high-resolution spatial context for those known phenomena and identifies specific genes associated with different layers and cell types, which is valuable. But it doesn't uncover a fundamentally new mechanism or target for intervention that wasn't already on the radar.
So its main value is as a reference, a resource.
Exactly. It's an outstanding resource.
Yeah.
If you are studying a specific gene implicated in aging, you can now use this atlas to see precisely which cell types express it, in which cortical layer, and how its expression changes with age in that spatial context. It's crucial infrastructure for validating hypotheses and designing future mechanistic studies.
But it's not the discovery paper itself. It's the detailed map that future discoverers will use.
Well put. It's foundational data, not the intervention blueprint.
Okay, that makes sense. So let's wrap up this week, October 13th to 19th. What's the overall picture?
I think the picture is solid, necessary, but mostly incremental science. It underscores just how hard it is to find those truly game-changing breakthroughs week after week.
The standout in terms of novelty was definitely the Fate gene editing platform for Progeria.
Yes, a real potential shift in strategy, targeting the farnesylation site itself, mutation agnostically. But as we discussed, it comes with major caveats. The organoid model limitations, the LNP delivery hurdles in vivo, and the critical need to demonstrate cardiovascular benefit. Still very early days.
Then the CMA paper in neurobiology gave us really valuable mechanistic insights, especially that striking sexual dimorphism.
Right, presynaptic versus postsynaptic remodeling in females versus males. That's a crucial finding for anyone developing drugs targeting synaptic function or CMA itself. But it also raised flags about the need for cognitive validation and the potential risks of long-term CMA activation.
The bone repair paper using PSLs looked clinically relevant.
Very relevant for orthopedics. The age and sex independence of the M2 polarization effect was the key strength there. But its application might be niche, currently limited to local bone repair.
Yeah.
We need to see if it works in other tissues.
And the big data papers, the multi-omics clock, and the spatial brain atlas.
Important resources. The clock paper highlighted the microbiome's central role and those intriguing sex-specific aging waves around midlife. The atlas gave us an unparalleled map of the aging cortex, but both are fundamentally observational correlation-based. They generate hypotheses and biomarkers, but not direct validated causal targets.
So the big challenge remains translation, doesn't it? Taking these specific findings, editing in a dish, liposomes in a bone defect, and turning them into safe, effective therapies that work systemically in a whole, complex, aging human.
That's the multi-billion-dollar question for the entire field, absolutely. Bridging that gap is where the real difficulty lies.
Which brings us to our final thought for you, our listeners, to consider this week. And it really ties together threads from both the CMA paper and that large multi-omics study.
It's this theme of the profound sex-specific nature of aging that keeps emerging.
We saw it in the brain, with CMA acting differently on pre- versus post-synaptic components in males and females.
And we saw it in the big cohort study, with those distinct molecular waves hitting females sharply around menopause, while males showed a delayed, more gradual change.
So the provocative question is this. If the fundamental molecular processes of aging, like metabolic shifts or synaptic decline, manifest so differently between sexes,
Then how long can we justify running generalized, non-sex stratified clinical trials in longevity research and expect them to be truly informative or scientifically valid?
are we potentially missing effective drugs because they only work in one sex and get washed out in a mixed trial? Or worse, are we developing drugs optimized for one sex that might even be detrimental to the other?
It really forces us to ask if treating aging as this single, monolithic, gender-neutral process is holding the field back.
Something definitely worth thinking about as you design your next experiments, analyze your data, and plan those crucial clinical trials.
Indeed, we'll be back next week to see what new signals emerge from the noise.